Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 62, and it's going live on June the 5th. From last week's heat wave, we now have thunderstorms, and the temperature has dropped by 10 degrees. We may be responsible for that because, on one of our other podcasts on hiking, this week's topic was heat stroke, although I do think that is a condition that we are going to see more of over the coming years. Last week our guests were John Friend, CEO of Casia Therapeutics, and we had a conversation with PureTech Health CEO Robert Line and Eric Elenko, President and co-founder of PureTech and acting CEO of Gallop Oncology. This time we're chatting with Revalesio President Greg Archambeau about a new way to treat strokes. Not heat stroke, that is, but it's a decent enough segue. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com. which is also the address you can use if you're interested in advertising with us, or sponsoring a podcast episode, or for event coverage. First, it's time to go through some of the headlines of the articles you may have missed over at deeptechdigest.com this past week. An anemia drug may have potential as a cancer treatment, lung cancer cells switch identity to resist treatment, and new peptides slip into cells to block hard-to-target proteins. A collaboration is hoping to advance a clinical trial platform for colorectal cancer. A new Tourette syndrome drug is showing promise in a phase 3 trial, and there were positive results from a kidney transplantation study. The first human SMUG1 structures reveal how cells repair DNA. There have been positive results from a regenerative cardiac patch platform, and also positive results from a C. diff vaccine trial. A new partnership is looking to accelerate next-generation brain treatments. A blood and biomarker testing service launched in Scotland for earlier Alzheimer's diagnosis, and you can read all of these and plenty more at deeptechdigest.com. All right, let's get to this week's interview. Revalesio is pioneering a novel drug class rooted in a surprising field: fluid physics. The company's focusing on strokes, among other things. Its therapy, RNS-60, uses a proprietary process to create an oxygen-supersaturated saline to boost mitochondrial biogenesis and ATP production. But let's ask the expert here, which isn't me. Revalesio President Greg Archambeau can tell us all about it. Hi Greg, great to have you on the podcast and talk about what you're doing. Could you give us a bit of an introduction to Revalesio?
Sure. Revalesio is a privately held company. We're supported by a couple of wealthy family offices, mainly based in the Pacific Northwest. And we're in the clinical stage with a drug for the treatment of ischemic stroke. And I think we've really caught the stroke community attention with the phase 2 data that we generated with our cytoprotective drug RNS-60. And looking forward to talking to you about it today.
Yeah, excellent. I wonder if maybe then we could talk about RNS-60 and what it is and the story behind its development and how it works.
So let's talk about what it is chemically first. So it's a oxygen supersaturated in sodium chloride, so saline solution. It's a simple product from a chemistry standpoint, but we've seen that the product has more of a biological response than just if you had an oxygenated saline solution. And that's due to the processing in which we saturate the saline with oxygen. And this is a technology that was generated with the idea of increasing crop yields in agriculture about 20 years ago. And we were generating data showing increases in yield and increases in stress resistance to disease or heat or maybe irrigating with brackish water. And Eric Russell, the founder of Revalesio at the time, just had the idea of what would happen if we applied this technology to human biology. And that's really how RNS60 was born. We had an opportunity to do some early work at Stanford, utilizing, again, our fluid physics process for saturating saline with oxygen. And we saw that the product had unique properties and almost changed the bioavailability of oxygen, where we saw an increase in mitochondrial stimulation within different cell types. And then we also saw kind of a dampening of inflammation in different inflammatory models that we looked at in the lab.
Sounds like an interesting start to the story. I mean, how do you make that connection between let's try it on humans? It seems like quite a leap.
It was, and I think, looking back, Eric Russell has a finance background. So it came from Russell Financial, the Russell 2000 Index. And Eric simply thought it's working in biology over here. What would happen over on the human biology side? And we were fortunate to have a connection with an immunologist at Stanford, and we were able to do some of that early work. But it's definitely not a product that was designed with a target in mind. It was something that we stumbled across when we were in an entirely different industry. And when we saw the results, we really dove deep into trying to develop an understanding of what RNS60 is doing at the cellular level. And for the next decade, just poured into foundational science about what is RNS60 beyond just oxygenated saline, but more importantly, what is the effect that it has? And so that led to a couple of dozen publications over the next decade as we made that decision to focus in on a potential therapeutic. Because the original goal of the Russell family when they founded Revalesio was to impact human health. They started off looking at agriculture because they thought we could solve some of the food security issues around the globe. So we're not focusing really in the Western nations, but going beyond that. But then seeing what the technology could do for human health, we've now pivoted, looking at the development of a therapeutic still in mind of impacting really human health globally. And, that's starting with stroke, which is a huge unmet medical need around the globe.
How did you settle on stroke as being the best way that this approach could help?
That's a great question. We did a lot of preclinical animal models in neurodegenerative diseases and neurological conditions, if you will. I'll talk about stroke there, and focused on both chronic and acute. So you might be aware that we've done some work in ALS, so a chronic neurodegenerative condition. We've run a phase 2 study in ALS with the support of the ALS Association. Got fantastic data there showing that we lowered or slowed the decline of breathing in people living with ALS. And then we chose stroke because, again, the huge unmet medical need, even with the best standard of care, today, if you have a large vessel occlusion ischemic stroke, you only have about a 40 to 45% chance of having a good outcome at 90 days. So there's significant need in that area. And we had done preclinical work there that showed RNS60 had tremendous promise.
And so, how does it actually help stroke victims?
Well, whether or not you're a stroke victim or some of the other neurodegenerative diseases that I've mentioned, RNS60 improves the structure and function of mitochondria when mitochondria is under stress. So mitochondria is important to all the cells in our body. It's the power plant of cells providing the energy that the cells need to perform work. And in a situation like stroke, where you have blockage of blood flow to a certain area of the brain, the neurons and other cells within the cerebral vascular system begin to suffer. And as that happens, you'll get mitochondrial dysfunction. And this can lead to a whole cascade of problems. And so what we've seen is that RNS60 seems to be supportive to those cells when they're under stress and allows the mitochondria to continue to function and produce the energy that the cells need to respond to that stress.
What are you doing in terms of clinical trials?
Yeah, so we designed a proof of concept study in large vessel occlusion. So these are major strokes. individuals that are receiving mechanical thrombectomy. That's where the clot is physically removed to reestablish blood flow to that area of the brain where it's been blocked. That's the best standard of care that we have in the field right now. So we designed a trial to begin dosing RNS-60 after the clot had been removed in these patients. Because to us, one of the problems in the past with stroke research is the selection of the patient. And companies have selected a kind of a wide range of individuals, creating a lot of noise in the data that's generated in small studies. So when we chose to select these individuals that were already having the clot removed, we thought that was the perfect scenario to study the administration of RNS-60 because blood flow has already been reestablished to that area of the brain. And then we begin to dose RNS-60 or placebo for 48 hours. And that was kind of the setup of the study. And we measured, this is one of the keys that really no one else in the field has been able to do. We measured the size of the stroke immediately after the clot had been removed. And then we measured it 48 hours later. This is the time window in which we dosed RNS-60. So we had a baseline for the size of the stroke at the start of treatment. And then we had an image at the end of treatment. And that's really what grabbed the attention of a lot of key opinion leaders in the field is we showed that RNS-60 reduced the growth of the stroke over those 48 hours by 50%, which is it's a massive number. Who doesn't want to rescue brain tissue?
Does the stage or the timing of treatment make any difference? I mean, could it have a positive effect on people that are at risk from stroke?
We haven't done any work on the preventative side. What we've been focusing on is the treatment of the stroke. Now, what we'd love to do, Jim, is to dose in the ambulance. If it's suspected that you are having a stroke, we'd love to get as early in the process as possible because we realize and the field realizes that time is really brain, and the earlier you can intervene, the better. The challenge with that is how do you execute a clinical study in that setting? And there have been some groups that have tried, and quite frankly, it can take 10, 12 years to run that type of study, and you only have a few dozen patients. So we made the choice, again, let's focus. And you also have the problem of variability within the patient criteria if you're enrolling that early. So we made the choice of let's try and get as homogeneous as a patient population as possible, realizing that treating after the clot has been removed might not capture the full effect or benefit of RNS-60, but based again on the preclinical work that we did, we knew that there was an opportunity to have an impact there. And let's move forward and potentially be in a position to file for market approval. Then we could look at administering RNS-60 earlier on within the process.
Because it's something completely different, does it have other applications potentially?
Absolutely. We've done work in acute myocardial infarction, heart attack. We rescue heart tissue in that model as well. We've done work in Alzheimer's and other neurodegenerative diseases. Stroke is our focus because the data is so overwhelmingly positive. And like I mentioned, 40 to 45% of individuals that get best standard of care and have a major stroke, it's only 40 to 45% of individuals that have a good outcome, I should say, at 90 days. In our study, we saw 63% of individuals have a good outcome. And if you're rescuing brain tissue in that earlier setting of the stroke, you would expect to see a benefit on individuals' independence. We very much like to get into to do another study in ALS. You talked about the other indications. The challenge there is that we've shown an impact on breathing for patients, and FDA would like to see would like to see a phase 3 trial designed around function. But when we've talked with individuals that are living with ALS, the number one thing they want is a cure. The number two thing they want is help breathing. And so we think we have exactly what patients want, but it doesn't quite fit with what the agency has communicated to us that they're looking for in a phase 3 trial.
This kind of general field of fluid physics, are there other unmet medical needs that can be addressed not necessarily by RNS-60, but this particular field?
Well, that's interesting. We think there's an opportunity to work with other biologically active gases like oxygen. We just chose to focus on oxygen first. We could look at administering RNS-60 not just through IV like we do with stroke, but through inhaled or other routes of administration. There's a lot of opportunities to explore with RNS-60, but again, our focus is on getting ischemic stroke through a phase 3 trial, hopefully approved, and then expanding the possibilities of what can be done with the technology that we've developed.
You mentioned earlier about acceptance and people taking notice of what you're doing. When you have something different like RNS-60 and it's outside of the norm, I guess, how difficult is it not only to break through with areas like funding, but also to get that kind of acceptance?
That's a great question. It has been challenging, and I think the key though is doing good science. So I mentioned it earlier. We spent about a decade just doing the fundamental work because we knew that there would be skeptics as it relates to this technology. Now, what we welcome those skeptics, actually, some of them have become now our greatest supporters because that's classic science. Come up with a hypothesis and challenge it. And so we've been hard at work over the last, I guess this goes back almost 2 decades of doing this foundational work. And I think one of the things that our investors have realized is that it's quite often when you have areas of unmet need, whether or not it's in pharmaceuticals or it's in some other industry, it's often that the solution comes from another industry and it comes from some space that the field is not focused on. And so we feel that that actually provides a bit of an advantage to us because RNS-60 is not tailor-made to impact a single receptor on a neuron. We think that that actually gives us an advantage in an area like stroke, where as you dissect the condition, there's about 6 different injuries that are going on at the same time. So you really want to have a drug that has a broad effect on human biology rather than more of a focused effect on, let's say, neurons. And again, we think that's one of the reasons why we saw such fantastic results in our phase 2 study is that broad effect that our drug has on mitochondrial function, whether or not you're talking about neural cells or glial cells or T cells.
I guess as well, 20 years ago, there was no such thing as AI assisting with clinical trial development. Has that become something that you've started to utilize, or are you beyond that?
When we designed our phase 2 study, we tried to fit into the standard of care as best as we could. And the feedback that we got from the clinics was this drug was so easy to implement. The protocol that you had put together fits perfectly within standard of care. So we haven't incorporated AI at this point in time in our clinical trial design. Our phase 3 is really going to be a duplicate of our phase 2. And as it relates to where we could go with fluid physics and maybe some of the other biological gases, that's an opportunity. We definitely use AI for other work, but as it relates to the design of our clinical trials, we really have the roadmap in front of us with what we executed on our phase 2.
Is that something that's going to be a help for what you're doing, the ability to produce at scale?
Yeah, so we've scaled the technology from our early laboratory experiments to now what we're doing in the clinic 3 times. So we're confident in our ability to scale manufacturing. Actually, our approach is taking what we have right now as a module and just adding additional modules. So our facility in Tacoma, Washington currently can handle our clinical trial needs and then could handle the first 3 years of RNS-60 being on the market, assuming that we generate the appropriate data and have a successful market authorization filing. So I think the other piece of your question, I think hinted on cost. And what we've seen with RNS-60 in this phase 2 study was a lot of benefits to the healthcare system as well, not just to the patient. If you received RNS-60 in this study, you left the hospital within 6 days. The placebo patients, it was 11. So 5 less days in the hospital is a tremendous amount of value. So we think there's an opportunity for hospitals, for the payers, for patients, and for Revalesio. There's a lot of value to share moving forward with the value that this product presents.
You talked at the beginning about the origins of the company and the agricultural benefit to the entire planet. Is this something that you're hoping will benefit everybody?
Well, definitely. I mean, that's the goal of the families that have invested in Revalesio, is to impact human health around the globe. As it relates to agriculture, that's something that we've put on hold. There's some cost factors there with regards to the equipment that currently don't make it economically viable to restart the agricultural work. But that could be something that's done in the future. Currently now we're 100% focused on the opportunities in front of us with ischemic stroke.
And speaking of that, I know it's very easy to overextend yourself with too much work and going in too many different directions, but are you working on any other things other than stroke?
Currently, there's a few things that are being explored preclinically, but again, focusing on this phase 3 study and executing it, I think will open up a lot of new opportunities for us. I mean, success in this setting could lead to more work in other areas of ischemia. If I go back to a heart attack. But again, it could also lead to proof of concept studies in chronic neurological conditions, or focusing in on areas of the body where mitochondrial concentration is the greatest. And so you could look at areas of like kidney disease, acute kidney disease. There's a lot of opportunities for us because of the broad effect that RNS60 has on multiple cell types because of its effect on mitochondrial structure and function.
For that next step with the phase 3, do you have timelines on that yet?
Yeah, so it take us about 2.5 years to execute that phase 3. Currently, we're raising funding to execute the study. We've selected 30 sites in the United States that would be our initial sites for the start of the study. So yeah, we're in the process of raising funds currently and eager to get the study started.
Is there anything else that you wanted to mention that we haven't gone over? It's definitely not the typical drug that we would necessarily be talking about, which is a good thing. It's nice to see things that are, completely different and having potential to shake up the industry and do good things at the same time. I guess that's the bottom line, is it's not just that it's different, is that it's making a difference. So
I think the key there, again, mentioning that sometimes solutions come from different industries or come from odd beginnings, if you will. But the key is doing good science. And I think that we've done that. In the area of stroke, we started off with a rodent model that showed we rescued 50% of the brain tissue post-stroke. And we moved into a primate model that showed the same thing. And then we did functional measurements then as well. And so when we moved into the phase 2 study, we were 2 for 2 on showing RNS60's effect on brain volume, on reducing brain volume loss. And when we finished the study, unblinded ourselves, I'd like to say we really weren't surprised to see that we didn't see a change in that effect. We saw a 50% reduction in brain volume loss in the human. So I think the key is to just continue to do good science. We're fortunate that we don't really need to make any changes when we move from our phase 2 study to our phase 3, which mitigates a lot of the risk in drug development when you have to tinker with the protocol as you move from proof of concept to a pivotal study. And fortunately, we're not in that situation. We're executing a study that we're confident we can execute and that we know the clinics enjoy being a part of, because it just plugs into their standard of care process for treating large vessel occlusion ischemic strokes.
yeah, that's always a major factor, as well as the fact that your partners in the clinical trials. It's a massive benefit to them as well. So they must be delighted by being able to be a part of this.
Yes, I think that's led to a lot of the enthusiasm that we see. And as you can imagine, the safety profile of RNS60 has really been great so far. They've told us this is an easy drug to explain to patients and to their families. And the safety profile that's been seen makes it even easier. So we're looking forward to decent enrollment rates once we get underway with the phase 3 study.
Anything that can help the many victims of stroke has to be a good thing. With every minute that passes in strokes, around 1.9 million neurons are destroyed, so time is hugely important. And let's hope that Revalesio's treatment is one of many being implemented in as many places as possible and as soon as possible. So that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please do feel free to contact us at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.